All right. Welcome to today's mathematical biology seminar. Today we are pleased to have Professor Leah Keshet, a professor at mathematics department at UBC. She'll be sharing with us her research patterns of cell signaling in one motile cell and many. Leah?
Thank you. Let me just get myself, okay, good. There's the first slide. Okay, so a lot of what I talk about was done obviously by students, postdocs, and colleagues, and I'll mention those as we go. And the first part of what I want to tell you is a little bit about things I learned way back in the late 70s when I was a graduate student with Lee Siegel at the Weizmann Institute. So he was working on self-organization of dictyostelium, of social amoeba that can come together to form a multicellular organism when they're starved and form all kinds of interesting structures, as well as spiral waves in their initial aggregation process. And what he was working on was basically PDEs that describe the initiation of this aggregation process. By the way, can you see my mouse okay?
Yes.
Okay, very good. Right, so like I mentioned, these sort of aggregates can produce spiral waves when the cells of these amoeba initially start to get together and form clusters, which later specialize into fruiting bodies. So I learned about that way back then. And then it turned out that actually spiral waves occur in all kinds of biological processes. So what I just described was the social amoeba, and it turns out that their chemoattractant is cyclic AMP. And the interaction between their secretion and chemotaxis moving up gradients of cyclic AMP leads to these spiral waves. You also have spiral waves in cardiac tissue, in various chemical systems, in MIND, which is a bacterial protein, and in other examples. And you'll see that spiral waves will come up in some of what I tell you as well. So I'm sure a lot of you, especially those of you who have worked with neurophysiology, know about the idea of excitability and the fact that, for example, in neurons, you've got these spreading self-excitation waves, and prototype model for these has been the Fitz-Nagumo model, which is a kind of elegant simplification of a much more complicated model by Hodgkin and Huxley. So just to remind you, the Fitz-Nagumo model is basically PDEs. The main action is in a PDE that has a kind of self-excitation term. And then there's a refractory variable, which operates on a slightly slower time scale and which basically damps out the excitation. And this simple system can give rise to traveling waves, as you can see down below. So in 1D, you see just a single traveling wave. And if you plot this on a space-time plot, also known as a kymograph, you basically see bands of these waves that travel across the domain. You can also simulate this in 2 dimensions, and lo and behold, you get spiral waves. So again, this is a system that is capable of generating such waves. So the basic takeaway message here is that an excitable system, or a system that has a kind of bistability that's coupled to slow negative feedback can give rise in certain conditions to these sort of dynamic patterns. Okay, now something else I learned way back in the late 70s when I was a student with Lee Siegel is the basic underpinning of the pattern formation theories that were initially developed by Alan Turing back in the 1950s, and then independently later developed by Hans Meinhardt and Geer, who did not know about Alan Turing's work at the time. And so again, these are PDEs with differences in this case, a pair of PDEs, differences in the rates of diffusion and certain kinds of nonlinear interactions that could give rise to standing wave patterns and that have been used to describe all kinds of biological patterns. So this just is a photo from back again in the same period, late 1970s, when I was a student with Lee. And this shows one of the workshops, an EMBO conference that Hans Meinhardt came to. And so you could see these were people that I knew and were my heroes at the time. Now, among pattern formation theory, again, another hero or another pioneer at the time, James Murray in the 1980s, wrote a paper about applying the Turing pattern formation type of PDE theory to animal coat patterns. So I'm sure most of you or many of you know about his books on mathematical biology, and you see examples of some of the patterns that he simulated at the time. And some of them are quite elaborate. I'm actually going to be focusing on one of the simpler variants of such a pattern, namely something that is merely polarized. So it has one level of activity or one type of coloration at one end of this animal, and another opposite end has the distinct different level of activity or color. By the way, I can mention that friends of mine, Avi Baran and Paul Monroe, actually took a picture of a goat like this that they had encountered, and it's actually featured prominently in Murray's book because they sent him such a photo. Let me also remark that patterns, of course, occur on many scales. And so I know at least some of you are familiar with vegetation patterns, which are not quite the same as reaction diffusion patterns, but have many of the same underlying local activation and long-range suppression features that the Turing patterns have. So these, in fact, occur ubiquitously at many scales. But I'm going to be focusing on the cellular level. And in my talk, I will mostly spend time on single cells and what happens inside those cells that polarizes them and allows them to become directional. But at the end of my talk, I will bridge to some of the collective cell behavior. Okay, so apologies if you've already seen this movie either in a talk I gave or online or in other talks. This is kind of a classic of cell motility models, and it's one of the few that really shows in great and lovely detail how our white blood cells can chase after bacteria and get rid of them. So what we're seeing here is a neutrophil, a white blood cell. It is able to move directionally in response to cues, such as proteins that bacteria shed. It can navigate between obstacles, and it's got, as you can see, a front end and a rear end. So it's directionally moving because it's polarized. And so that's really what one of my main initial questions is: how do cells undergo this polarization? And what we've seen now is, in this last slide, we've seen a view from the top down. And so really the cell is three-dimensional, but in a lot of what I say today, I'll think of the cell as flattened. So you'll either see it as 2D or even as a 1D reduction for the purpose of simplifying for mathematical tractability. And when it's in 1D, so it's kind of like a diameter of the cell, then the two ends are the ends of the cell, and they would be sealed. So I would use what's called Neumann boundary conditions, because nothing can leak out. So just to be clear what my domain is going to be. Okay, now let me tell you a little bit about what's going on in the cell to allow it to move. So inside the cell, there are structural proteins called actin, and they form filaments that extend at their ends. And those ends are basically pointed toward what becomes the front edge of the cell. And as new monomers are added onto these biological polymers, that allows the cell to push out or protrude. Now, the back of the cell has to keep up, and the way that happens is that there are little actin fibers at the back that are attached to myosin motors, causing the rear of the cell to become contracted to pull it up so that the cell doesn't just spread out and become distended. And that's what's allowing the cell to migrate directionally. But to control that and to enable the actin to polymerize at the right place and the myosin to work at the right place, there's this huge internal regulatory system that talks to the actin, bringing the information from stimuli outside the cell, which would be on the top of the diagram, via receptors on the cell membrane and through a variety of internal protein transduction networks down into a system that controls the actin cytoskeleton. And I'm going to focus attention on a key signaling hub, which is called the small GTPases, because those are known to be sort of the primary components that orchestrate the ability of the cell to polarize. And so there are several different kinds of so-called GTPases that are in this central hub, and some of them link directly to actin polymerization, and others activate the myosin motors. So you can think of it as some of these are going to be responsible for the front protrusion of the cell, and others for the retraction of the rear. And so it will be important for them to also become localized inside the cell so that they will only allow actin to polymerize at the front and only allow myosin to retract at the back, roughly speaking. So, of course, there are a lot of fine details that I'm going to gloss over just to give you an idea of what's going on. So there's something important that I need to tell you about each of these GTPases. Basically, they are protein switches. So they act like on-off switches that are either in an active form where they have an effect, or an inactive form where they're basically doing nothing other than becoming a pool available for activation. And interestingly, the active GTPases are bound to the cell membrane. And when they get inactivated, they come off the membrane and are then inside the fluid part of the cell. And interestingly, this means that the rates at which they can diffuse across the cell is going to be quite different, because the active forms in the membrane are in a highly viscous environment, so they diffuse slowly relative to the inactive proteins, which are in the fluid part, and so diffuse quite fast. Okay, so each of these proteins can be seen to also spatially localize and in a dynamic way. So here's a movie from the lab of Orion Weiner, where basically he took one of these white blood cells and stimulated it. And you can see that this wave of activity of one of these GTPases, the RAC, starts to invade into the cell. So the cell outline is roughly around here. And you can see here, and also in the still time frame, the fact that the activity, the bright portion, which is the active RAC, first invades inwards and then relaxes back to a kind of polar distribution. And notice that this happens very quickly on a time scale of seconds, which is much faster than the rate or the time it needed for proteins to be made inside a cell for protein synthesis. Okay, so briefly, the signaling proteins that I've told you about, they're what determine the localization of actin and myosin. And they have to form an initial polarity map in order to do their thing. So they respond to external cues and they form a simple front-back pattern, as I've shown you in the movie. So the same kind of pattern that we basically saw on that goat, this is a real goat, it's not been painted, occurs in the cell as far as the activity pattern of, let's say, RAC or Rho, these regulatory proteins. Okay, so now I'm going to reduce the problem of explaining how cells polarize by accounting for the spatial redistribution of these signaling proteins, which is really a pattern formation problem in the style of Turing and Murray and all these other people who've worked on pattern formation. And again, let me emphasize the geometry. So the cell, we've flattened it so that it's a sheet, and it's basically a membrane all around with a fluid interior. And we're abstracting that here as a collection of molecules, which are regulatory proteins, some attached to the membrane, some are in the fluid, but we won't consider different compartments. Instead, we're just going to assign them distinct rates of diffusion and look at the whole thing as a one-dimensional thin strip and try and understand how patterns of localization can form. So that's the problem that was handled by two former group members, Yoichiro Mori, a postdoc, and Sasha Zhilkin, who did her PhD with me. And so what they did was they simplified this vastly complicated network by looking at just one of these proteins, Rho or CDC42 or RAC, considering just its active and inactive forms as shown in this little icon on a 1D geometry, as I've explained, and put together a set of PDEs that could model this redistribution and on-off interaction. And in order to get anything interesting to happen, some kind of feedback or nonlinearity was essential. So the kinetics in the PDE here are, there's a kind of Hill function for mutual self-activation. There's a kind of decay rate, and there's mass conservation because, like I said before, we don't have any synthesis of the proteins on the relevant time scale. We merely have them exchanging their state between active and inactive. And so with that simple 1D PDE model, they were already able to get something interesting. And here again is the chymograph, where what you see is that from an initial stimulus at one end of the cell, you see that some kind of final pattern emerges where part of the cell is in the highly active form and the rest of the cell is not. And now the main ingredients, because the model is simple enough that it can be analyzed mathematically, it turned out that the main ingredients are the conservation, the mass conservation, pardon me, the distinct rates of diffusion, and the positive feedback. Just to contrast, a similar kind of function in a single non-conserved system would give rise to a traveling wave that simply goes right through the domain, whereas what we see in the analogous mass-conserved system is what we call this wave pinning. The wave goes into the domain and then it stalls in the middle. And we can see that also in these two simulations, where in one case, at the end, everything is uniform. That's the single reaction diffusion equation. But with the wave pinning mass conserved version, the wave will freeze. And that's important because it gives us this ability to explain a stable polar pattern, whereas this system does not. Okay, so so much for that simple PDE model. Now I can mention that because it's analyzable and it's easy enough to understand and to simulate, you can get a lot of insight into how it behaves. So it's not sensitive to noise. The stimulus has to be large enough in order for it to become go into a polar pattern. The same idea works in 1 dimension, 2 or 3 dimensions, and I already said that it's simple to analyze. It has other kind of interesting features. So, for example, if you simulate the same PDE model on a 2-dimensional domain, then it turns out that the highly active parts of the pattern respond to the domain geometry. So here you see examples in both an oval and a somewhat irregular domain, where you can see that the pattern gets trapped by highly curved portions of the domain.
Leah?
Yes?
We have a question in Q&A
Okay,
session.
go ahead. Yes.
Could you please remind us what the boundary conditions were for
Yes.
the 1D model that you
Yes.
showed on the previous
The
slide?
boundary conditions for all of what I described are going to be Neumann, no flux boundaries. So nothing escapes from the boundary. And it turns out, by the way, that this has a link to the long-time dynamics of the mass-constrained Allen-Kahn equations in a Neumann setting, Neumann boundaries, where the curvature of the interface affects the evolution of the pattern. So there's some interesting math that is in the background. Are there any other questions before I continue? Nothing? Okay.
Thank you.
Let me go on then. So it turns out that there's a link to some interesting mathematics. So, for example, in this paper by Alikakos and others from more than 20 years ago, they basically showed that these kind of systems, the Allen-Kahn type of system, will first of all undergo coarsening, then be attracted to the boundary, and then this little droplet would move towards the highly curved and be trapped by the parts of the domain that are highly, that have a high curvature. Now, this was done for small droplets because then they could use the techniques of asymptotics and various other variational techniques. But it turns out that similar things are at work here in the background. Now, we've looked also at how this same simple system works on a deforming domain. And that was done by a former postdoc, Lizanne Wenz, and an undergrad student, Zachary Pellegrin, who coded up this type of simulation in a deforming boundary. This is a bit more challenging because of the fact that solving a reaction diffusion equation on a deforming boundary can give rise to all kinds of issues as the boundary deforms. But you can see that, for example, from 4 different initial conditions, you get basically evolution of shapes that look somewhat like a cell or like a cell that's got protrusions. And there's a lovely paper by Brian Camley that some of you might like to read, where he uses a very similar kind of internal cell reaction diffusion mechanism, but he does this on a deforming cell using the phase field method, which is used by many of the physicists, and they discuss this instability of cells that either have a straight trajectory or that turn and move around in a circular trajectory, depending on the relative tension at the edge compared to the outward forces of the protrusion. So that's a cute little connection in his work. I'll mention that this was also simulated, same model in 3D with colleague Anotida Mazvamuse. And by the way, Anotida recently joined us here at the University of British Columbia. So he's a wonderful new colleague that I'm enjoying working with. All right, so I'll just briefly mention the fact that when we deal with these models, we like to have some shortcut mathematical analysis methods. So one of these, which was called the local perturbation analysis, is something you may want to check up if you want. And essentially, it's a shortcut method that answers just one question. If you have a reaction diffusion system and you stimulate it with a small pulse, will that pulse decay or will it grow? And how does it depend on the pulse magnitude, the amplitude of the pulse? And it turns out that that allows one to approximate the PDEs with ODEs just to answer that question. And then you can use ODE bifurcation analysis to go after and understand when polarization can happen. And the reason why that method is useful is it works equally well for the simple one GTPase model I've been talking about, and for the more elaborate circuits that one might be interested in. Okay, so much for the one single polarization model. And now let me talk a little bit about some of the variants we've looked at. So here's the original model, and in the background is the fully complex version. And we've looked at all kinds of more and less complicated variants. I won't talk about the complicated ones too much today, except for mentioning this one briefly later. But I do want to discuss one in which an excitable system shows up, and spiral waves as well. So here, all we've done is we've added one more ingredient to our core signaling model. Namely, we've added the fact that the GTPase provokes or enhances actin polymerization, but also that the actin polymerization inactivates that GTPase. So there's negative feedback added to the model. And you can think of this as slow negative feedback working on an inherently bistable system. And that's really why we're going to be able to see excitable behavior. So here are some of the examples in chymograph form for the top two rows of patterns that you get merely by adding this F-actin feedback. So you can see that you begin to get oscillations. They can be pulses. They can be traveling pulses that move and bounce across the domain. And in 2D, you get spiral waves. And this is work by both Bill Holmes and Yu Liu, who were a former postdoc and a former PhD student. So interestingly, spiral waves are actually seen in real cells. So here's an example of a movie of endothelial cells, cells that line up our blood vessels. And what's seen here is a stain for actin. And you can see that there's some beginnings of spiral wave happening. And as that wave hits the edge of the cell, now you'll see that it pushes out that edge. So here's a simulation by Lizanne of something similar, where she's simulated a reaction diffusion system precisely as shown in the icon up here in one of these deforming domains. And you see that it has this spiral wave in the interior, and it pushes out the cell boundary. Here's another lovely example from real biology, from the work of Bill Bement and George van Dassau, of actin waves in real cells. So this happens to be an egg cell. It's an egg cell of a starfish. And similar kinds of waves also happen in frog eggs. And here what you're seeing is actin in red, and one of these signaling proteins, one of these GTPases in green. And the chymograph is shown on the right-hand side. So it's nice to see that these things actually occur biologically.
Leah,
Questions?
I have a very biology question.
Yes.
So I understand this F-actin dynamics has some role in motile cells, but what is it doing in the egg cells?
Aha, okay. So in the egg cell, it's actually going to form the equatorial ring that will constrict in cytokinesis. So we're not seeing that in this cell because some of the proteins were downregulated to prevent that the formation of that equatorial ring. But in an actual egg cell, the row is going to basically eventually form a ring around the cell equator, create sites for assembly of actin and myosin, and that ring will constrict like a purse string and pinch off and form two daughter cells. Does that help?
Why does it need to be spiral pattern?
Oh, no, it doesn't. Okay, good question. So these spiral patterns do not occur in the natural egg that divides, but they can occur if certain proteins are tweaked so that the waves are not focused on the equator, but rather they can form this dynamics all over the egg. Yeah.
Got it. Thank you.
Thank you for the question. Okay, so just recently, we've done, I've been working with a few people who've been interested in this area, and we've been writing a review paper that we just submitted to eLife with a review of actin waves in a variety of species, including social amoeba and frog eggs. And it turns out that a lot of people have put together models as well as done experiments in a variety of cells. And it turns out also that many of those models share some kind of key, let's say, topological structure or graphical structure. So there are models for white blood cell neutrophil waves, for waves in the frog eggs and nematode worms and starfish, for dictyostelium. And you can see that a lot of them have in common this idea of an active variable, a reservoir, positive feedback, and a kind of negative feedback cycle. So we're thinking that there's some kind of unifying principle, and we're working on some of the mathematical analysis of this system. Let's see, how am I doing for time? I still have, what, something like 10 minutes?
Yeah,
Is that
50
right?
minutes, yeah.
Okay. So I want to gradually segue to multiple cells, but let me do so via mention of some experiments that we've been involved in. Now, there's a variety of different experiments that we, or collaborators that we worked with on models where they did the experiments. But today I'm really only going to focus on one as far as the single cell models, and that is the cell repolarization. So the basic idea is I'm going to try and describe what has to happen if a cell that's already polarized is going to be able to change its orientation and repolarize, to reverse its direction of motion or change its polarity. So it turns out that we looked at this together with a group headed by Andrew Levchenko back a good, well over 10 years ago. And his student, Ben Lin, at the time was looking at cells in microfluidic channels. And he was piping in gradients of a small molecule that caused the cell to develop this internal polarization and to polarize. So the external gradient caused cell polarization, and he examined different initial cell states, either cells that were initially polarized, not polar, polarized in the wrong direction, etc. And he showed that, in fact, the external gradient gives rise to this internal gradient of GTPase. In this case, it's RAC, but you can see that, in fact, there's high RAC activity that is caused by the cell, by the external gradient. So then my postdoc, Bill Holmes, put together a model for this internal signaling. You can see the model is quite a bit more complicated than the one I described, but interestingly, it had very similar kind of bifurcation signature when it came to this shortcut method of local perturbation analysis. Again, I won't go through the details just because there's for time reasons, but this has all been published, and I can send you links to papers if anyone's interested. So it turns out that the model shown down here accounts for the data for the way that cells respond. So different gradients give rise to different response times, for instance. And also it explains the way that cells can repolarize. But more recently, there have been interesting experiments in the past decade or so that now use optogenetics. And so what they do is they stimulate a cell with light. They take a cell that's moving in one direction, stimulate it with light, and cause it to reverse its polarization, because the light essentially activates, let's say, RAC at the rear of the cell instead of at the front. And what you see is that, in fact, the cell will turn around and reverse its polarity. And so together with former postdoc Andreas, who's now a faculty member in Amherst, Massachusetts, we looked at how to explain the cell repolarization based on the simple circuit I just described and one that's just a little bit more complicated. And in particular, the question we asked was, how much signal intensity is needed in order to get the cell to actually reverse its polarity, as distinct from losing polarity or simply relaxing back to the same polar state. And Andreas was able to put together a rather complex full PDE bifurcation analysis to answer this question. And so he was able to show that you need to have a signal intensity of sufficient strength, depending on the internal cell parameters, in order to fall into a regime where you guarantee that you can repolarize. And otherwise, you either lose polarity or just fall back to the same polar state. And these are just some simulations in 1D moving domains that show the same idea, where some stimuli, here's the second, this first stimuli polarizes the cell, the second one, in this case, repolarizes it. In the other case here, it completely causes the cell to lose polarity, and here the cell just continues in the same direction. So that was something we did recently, and also applied it to collisions between cells, where here we're seeing the internal pattern and how it changes based on dynamics of the cell collision. Okay, finally, in the last 5 minutes or so, I just want to, again, bridge to what we've been doing more recently in multiple collective cell behavior and in many cells. So I'll very briefly describe two projects, and essentially you'll see that in order to go from one cell to a collection of many in the tissue, we basically simplify the cell. So we're not looking at spatial distributions within cells. We're not looking at PDEs inside cells. We're adding cell-cell interactions. We're adding how the internal cell state affects migration and adhesion, but inside the cell, we're modeling things with ODEs. So example one, there was work by Touboul et al. That appeared in Science about 5 years ago, where this is synthetic biology, where they took cells and reconfigured their internal gene regulatory systems to control their adhesion. And they showed that they could get those cells to form clusters that kind of were self-organized. And one of my students who took a course from me at the time essentially took their genetic circuit that they had engineered, figured out how to model it. It's basically a delta notch kind of a system, and implemented it in a package called Morpheus, which is a multi-scale simulation package. And so that package enables one to link what happens inside single cells to what happens in for example, a two-dimensional cluster. So here what we see is initially cells that are not well organized, eventually forming internal states, adhering to each other in such a way that they form a three-layered cluster. A second example comes from work by Jin-Suk Park and Andrei Levchenko, again, same lab as before, and has to do with wound healing and the transition between an epithelium and a mesenchymal state. So what you're seeing here is a movie where you have many, many cells with an edge that migrates outwards, and occasionally, under some cases, cells break away from the sheet. So what they did is they grew these cells on patterns, and they saw that the cells lost adhesions and became highly motile. In other words, they underwent what's called EMT, the transition between being a sheet and individual cells, epithelial mesenchymal transition. And they also characterize the internal signaling that gives rise to that kind of behavior. By the way, RAC is the same kind of GTPase that I discussed earlier. And there's an internal mechanosensitive protein that somehow sits in the middle, controlling adhesion of the cells, as well as motility. So one of my students who did a master's at the time modeled this circuit and implemented it inside individual cells in the same Morpheus package to understand the way that this wound healing takes place. So again, each of these cells has a set of ODEs, and those ODEs then are linked to cell-cell adhesion as well as cell motility. And we see this type of a dynamic as the time goes by. And in fact, what we're seeing is relatively close to what's seen experimentally in this type of wound healing. We also, our simulations recapitulate the kind of trajectories that they found experimentally, as well as the behavior such as cell speed, as a function of distance from the cell edge. So the data from the paper, the experiments are on the right here, and the simulation on the left. Okay, I'm basically out of time. And so let me just summarize very briefly that as far as single cells, I hope I've persuaded you that starting from a very complicated biological system, we simplified it greatly to be able to highlight some of the key components and necessary interactions that allowed for internal cell polarization to take place. And that tiny 2 PDE model was simple enough to analyze mathematically. I didn't talk about that analysis here, but we've written papers on it. And then gradually we've been adding components one at a time to see how they work together to find some of the dynamic patterns occurring in cells, including actin waves. And as far as many cells, we capture the internal states of a cell, not by spatially distributed, but rather by time behavior of ODEs for the time being. We then include the interactions such as adhesion, cell growth, cell death, and so on. And we found that it's really useful to use packages like Morpheus and CompuCell 3D to visualize and carry out these simulations. And I must say that using these packages also allows for many very nice projects that are suitable for beginners, including undergrads that have worked with me. Okay, so with that, I will thank you for your attention, and I'll be happy to take questions.
Thank you, Leah. Do you have any questions? Oh, yes, we have a question. Oh, yeah. Yes, saying thank you. I have a very, I guess, basic question. So for the multicellular system, you used ODE for each cell.
Yeah.
What if you consider spatial pattern in each cell? Would it make qualitative difference, or?
It would surely make a qualitative difference. The main issue there is that currently there are no existing packages that allow you to easily do that for multiple cells. So it is done by custom simulations by some groups where they, for each cell, they'll have internal dynamics. At the moment, neither Morpheus nor CompuCell 3D nor Chaste are able to, Chaste is another one of these free platforms, are able to do that. It would be more intensive of resources, but it would also allow you to do other things like actually look at cell polarization and how it affects the interaction of cells in a tissue. So cells may interact differently with those that are in front of them than with those that are behind them, right? That may depend also on their interior distribution of signaling proteins. So it could lead to qualitative differences indeed, yes.
I see. We have a question, but I guess, can you share a few words about the results of the other more complex model with capping protein and ARP?
Sure. So let me go back. Oops, sorry, let me first go back to right. So this model here This is something that was done jointly with Stan Murray and a few other people, two of which were students. And it was also in a motile cell, in a moving cell. I don't have a slide for that here. I can certainly send references. But interestingly, it showed similar kinds of overall behavior. So it showed the fact that an initially round cell could be stimulated by a large enough stimulus to polarize so that CDC42 and RAC were at the front, where they could then give rise to ARP23, which then gives rise to actin filaments, which push out the edge, and Rho would be at the back, and that was due to the mutual inhibition of these two things, and that would cause contraction of the back. And indeed, we found that cells begin to polarize and in fact move in a steady state polar shape, and they could be induced to turn by stimuli that reorient them. So it had many of the similar features to what we discovered with the simpler variant. But when we worked on this, we still were a little bit grappling in the dark as to what are the very minimal requirements. And that's essentially why we eventually looked at the smaller models. Does that help? I don't know if I answered the question there.
So it says, interesting, would appreciate the references.
Sure.
So the result was size dependent.
The result, we have shown that result is size dependent. In fact, we've shown that for the small model that we could analyze completely. I don't have the bifurcation diagram here, but we showed that indeed several things had to be true. The size of the cell could not be too big or too small. And the total amount of regulatory protein had to also be within some limit. And the reason for that is the polar pattern forms as a result of depletion of the inactive form and the concentration of the active form at the front. So if there's too much total regulatory protein, then the inactive form never gets depleted. So the wave will just go right through the cell. So yes, indeed, polarity can only happen within certain conditions.
Thank you. Do you have more questions? If not, we also run out of time. So
Yes.
let's thank Leah again.
Thank you very much.
Thank you so much.
Take care.
Thank you very much, Leah.
Thank you. Hope to see you in person.
Likewise. Can you send me a link once you have the recording?
Yes, we'll do that.
Okay. Take care.
Have a
Thank
wonderful
you.
day.
Bye.
Bye.